Okay, now let's talk about the cells and biotechnology. There are different cells that are available out there. Of course, human cells that we have. Besides that, there are bacterial cells. We consider them as biobuttons, right? Besides the bacterial cell, there are the unicellular organisms, especially eukaryotic cells, like algae, green algae kind of thing. And then there are fungi made up with yeast and molds, and mushrooms. And then there are plant cells. And then we are here in this group, animal cells. So there are so many different types of cells that are out there, and why and how they are important for biotechnology. Think about it. In our traditional old definition of biotechnology, use of living cells and living system to produce something. That was the definition of biotechnology. If you think about that, maybe your dairy production, like milk and cheese, butter, you use a bunch of bacterial cells during that process, like lactobacillus will help you to make those. Not only that, there are different types of examples, like making wine or beer. You use different types of bacterial cells in there, mostly yeast. So classical example, yes, we have been using a bunch of different cells in our production. So that's a traditional biotechnology. But how about the modern biotechnology? How are we using cells for a more sophisticated way of making products? Think about the pharmaceutical industry. Right, so those pharmaceutical products, injectable products, a lot of them are made up with proteins. Think about vaccines of different kinds, hormones, and maybe antibodies. Those are all protein-based drugs. So we use cells to make those protein-based drugs. And in order for us to do that, yes, we have to genetically engineer those cells, and those cells will make the protein for us. Okay. So another name for those genetically engineered cells or organisms like animals and plants, we call them transgenic. Transgenic means transfer of genetic elements. That's what transgenic means. The same words as genetically engineered. Okay. This picture shows you what kind of animal cells, growing under the probably petri dish or maybe in vitro condition. Okay. So what kind of cell types are used a lot in the biotech industry? Okay. Yes, bacterial cells are used a lot. Think about insulin, for example. Eli Lilly has been using E. coli as the main cell type to produce insulin proteins. Okay, so yes, it's cheaper to use bacterial cells to grow up, and then it's cheaper to break open the cells and make the proteins. But there's a problem of using bacterial cells in making proteins. Any proteins that are large, and also any proteins that have complex structure, then bacterial cells cannot make those. They don't know how to make those structures and proteins. And think about chemical modification of your protein. We call that glycosylation. Glycosylation means a lot of your proteins are attaching the sugar groups at the end. So that's glycosylation. So a lot of human proteins, the receptor proteins, antibodies, have glycosylation there. If your proteins glycosylated, E. coli cells do not know how to make those glycosylated proteins. So you cannot use E. coli cells using those large glycosylated protein. Then you have to use some other cell lines. Another cell line the industry has been using a lot, fungi-based cell line like yeast cells. So a good example is Saccharomyces cerevisiae. That's a baker's yeast that we used a lot in our bread making. Okay, and then another yeast cell line is Pichia pastoris. Yes, Pichia pastoris, it can be used a lot because it grows well under Iraq's condition. So a lot of vaccines are manufactured using these yeast cell lines, like hepatitis B vaccines and things like that. So yes, yeast cells are growing well compared to bacterial cells. It's a little cheaper. It's a little slower growing. So bacterial cells, if we grow them, they grow very fast. Every 20 minutes they double. Yeast cells grow relatively fast, but not as fast as bacterial cells because they double every 2 hours. There's a little difference in the growth rate. How about animal cells? I told you large complex proteins like that are glycosylated, highly chemically modified, cannot be made within E. coli cells. Yeast cells as well, they cannot do the glycosylation completely. Then if that's the case, what is the choice of cell lines? And then you have to use cell lines that are similar to human cell lines, such as what? Mammalian cell line. Examples are Chinese hamster ovary cells. Chinese hamster is what? It's a mammalian mammal. Okay, so they are very similar, like us. We are mammal, Chinese hamsters are mammal. So the cell lines that are developed from Chinese hamster can make human proteins that are large and very complex. That's why many biotech companies, pharmaceutical companies, are using CHO cells to make human proteins. Okay, because they can make large and complex proteins that are glycosylated. Okay. When you grow up cells, you need to know how to provide the correct environment. Of course, cells need to eat and they need to be kept at a good temperature condition, and they need to breathe as well. They need to have oxygen for their cellular respiration. So all these conditions should be met, like water. They should have enough water, food, oxygen, right temperature, pH environment as well. So you need to provide the optimum best condition. Then cells can grow, they'll duplicate, right? And the cell division, reproduction is your goal. So you'll have more cells that can make your product for you. Okay, so you have to keep them happy. That's basically what it is. And then how do they grow when they are happy? Typically cells show you this kind of growth phases. Okay, when cells are first introduced to a new growth medium that's fresh, they don't grow up right away. They get settled and get adapted to the environment, and they don't double. So we consider them in a lag phase. Okay, they try to get adjusted to a new environment, and once they are adjusted, they start to reproduce. So this is a reproduction phase. We call the log phase cells, log phase, growth phase. And then finally, when they run out of space and nutrients, they don't double anymore. That's a stationary phase. If they are kept in the stationary phase a long time, and then they'll die off. There's another phase called death phase after that. Okay, remember, lag phase, log phase, and stationary phase. And if you're growing cells, then where should you keep those cells at? Of course, you need to keep them at the log phase, especially at the late log phase. That's when cells will be happy, healthy, and they continuously divide. Why at the late log phase? That's the time when you have maximum amount of cells. If they go into stationary phase, that's not good because they are overcrowded, they are not happy, and they start to release toxins and enzymes that will chew up your protein. So you definitely don't want to keep your cells in the stationary phase. Log phase is the healthiest phase, and you will have many cells if you keep your cells at the late log phase. Okay, that's your goal. So take a look at the explanation of each phase. Lag phase, initially, they are getting adjusted to the new environment. Log phase is when they are very active. They are continuously dividing. You want to keep them happy in the log phase, and they'll eat and reproduce. And stationary phase, when they're not happy, they will start to fight by releasing toxins. They kill up each other. Not good. Okay. And then, so in the biotech industry, yes, you will be genetically engineer those cells, make transgenic cells, and you will grow them kind of optimum condition. Only those cells that you need will be grown aseptically, right? So we call that process, cell growing process, as upstream processing. So you start out with a small 1 mil cell ampule that's frozen. You throw it out and start to bring the cells back to the life. And cells will go through the growth phases, lag phase, log phase, and stationary phase. But you want to keep them in the stationary phase continuously, and they will double, and you will get a lot more cells made. And then they'll eventually run out of space and nutrients, and you have to pass those cells to a larger fresh medium. passing, continuous passing to expand the cell number, cell culture. That's the upstream process until you reach the maximum condition with the maximum number of cells. And when that happens, you're going to harvest the cells, and then cells will make the proteins for you. And what you need is the protein, not the cells, right? During the downstream process, you'll try to purify the protein from the cells or medium that you don't want. That's the purification process. Once you captured, purified your API, then you're going to formulate, fill them in a vial, and then you're going to label it and package them. So that's the drug product manufacturing. So that happens, those steps happen during the drug manufacturing processes. Okay, so upstream and downstream process, which are called drug substance manufacturing, are all important, as well as the drug product manufacturing. And during those processes, you have to use documentation, of course. So during that manufacturing process, product batch record is really important. Master batch record should be kept within the QA department, but working batch record will be used continuously, so it will follow your product processing, and then every step you will use SOPs to make sure everything goes the same way according to the specification. Documentation is important, right? And then bioreactors are the vessels where you're going to grow your cells at. And bioreactors are unique because they provide the real-time monitoring of all those necessary parameters like a pH, oxygen level, CO2 level, and nutrients and temperature. All those things are monitored, controlled, so you can provide more needed elements in the cell growth. Okay, so bioreactor is really important. And bioreactors come in different shape, different sizes, as well as different materials. There are stainless steel bioreactors. They are reusable, and glass bioreactors that are smaller. And then there are plastic bioreactors that are tend to be used as a single-use bioreactors. And then, yeah, so you need to understand different types of bioreactors are used, and you need to know how to make those and set up those bioreactors for your cell growth. And then cell suspension. Cells come in different conditions. Some cells tend to be suspended, meaning they can float, and if you provide the right condition, they'll float, just like E. coli cells or yeast cells. Animal cells can be floating. However, the naturally happening animal cells are not floating or suspending like your cells on your skin. They tend to stick together, so they form layer, normally single layer. So we call them adherent cell. Adherent meaning sticking on a surface. So you may be growing both cell lines, adherent cell lines or suspension cell lines. Of course, suspension cell lines are much easier to grow because they'll be floating in the cell. You can just count the number of cell concentration and make them under the late log phase. But adherent cells, you need to provide the enough space, surface for them to stick. So you need to be careful. So if they run out of the space, and they tend to go through the stationary phase, which is not good. Okay. And then when you grow cells, especially suspension cells, your bioreactor can come in as batch bioreactor, or fat batch bioreactor, or continuous bioreactor. Okay. Or there are different types. You need to understand the difference among them. So batch is, you set up your bioreactor with a defined volume. So you inoculate the cell within the defined volume, and you grow until the cells reach the late log phase cell. That's it. That's the fat batch culture. Fat batch is you set up your bioreactor with little less than the maximum volume, and you continuously feed nutrients, fresh nutrients, so cells can grow more. That's a fat batch. And continuous bioreactor is you set up your bioreactor with less volume, and so when cells grow, you can feed them with the new nutrients. Same time, you can collect some cells from that the bioreactor. So you continuously feed the cells and collect the cells, feed the cells, collect the cells. So you can extend the culture condition for a long time. That's a continuous bioreactor. Okay, they are all kind of different, the way you monitor. Okay.